So the process of translation is the process of turning the RNA transcript that was produced during transcription, turning that into a protein. And we'll see the process here. First, want to introduce you to a little bit of the concept, kind of how this fits in with genetic inheritance. So when we have a phenotype, let's say you have an interesting phenotype, if it's the result of a gene, a difference in genes, an allele, then that would result in producing potentially different kinds of proteins. So here's an example from humans, a nice interesting example. Uh, there's a proportion of people who have a different version of this protein here. This is a protein called TAAR5, and it's involved in scent perception. And you can have a different version of this protein that causes you to have a different intensity of smell reception. So this is from a study where they were determining that some individuals homozygous for a particular allele were not really detecting, I don't know what's going on with these 2 points, but on the whole, there's lower detection in individuals that have this allele. They were smelling a fish sample, a sample of fish smell. Kind of like this example because it's a kind of a weird out there phenotype. So some people are genetically less turned off by stinky fish smell, and there's a genetic cause to this. And it comes down to how their scent receptor protein is made. So on the topic of translation, how is a protein made? Where do we get this protein from? Uh, so as we've talked before, of course, there are genes made up of DNA, and the DNA from those genes contains the instructions for making proteins. This is the most typical way that we think about genetics and the function of genes is that they are producing protein. So there's instructions buried in the DNA for how to make a protein, and that's a big part of the topic we're going to cover here. So I went and grabbed, this is the output from the human genome for how to make this TAAR5 protein. This is available from a resource called GenBank. And uh, based on the fact that DNA has these 4 nucleotides, right, A, C, G, and T, uh, it's kind of takes a special ability for us to understand, got to have a certain knowledge to understand how these letters that are not terribly variable, how the letter code ends up becoming the code for making a protein. So some of this we'll cover in the topic on transcription, but I want to kind of dive in just here and talk about translation and cover that as a concept all on its own first. So you can have different versions of genes that produce different versions of proteins. So buried in here, this is the DNA sequence, and in there are some parts that are known as codons. And the codon region is the region that most specifically contains the instructions for what parts of the protein to put together. So you wouldn't necessarily, just looking at this series of letters, you wouldn't necessarily know where to find these start codons, stop codons. But we're going to maybe kind of explore how to find them. So the start codon and stop codon, as we'll see, these are specific sets of 3 nucleotides, and they contain the instructions for starting to make a protein and ending the protein sequence. So something you might have picked up on is that this sequence here shows DNA nucleotides, and DNA is not the molecule that's involved immediately in making proteins. It's actually the RNA. So the process of transcription that we will talk about in a separate lecture, transcription turns this DNA molecule into RNA. The only thing I did here was I switched the T nucleotides, which are a DNA nucleotide, I switched those into U nucleotides, which is an RNA. So DNA goes ACGT, RNA goes ACGU. So it got a little bit, changed the orientation a little bit here, but stop codon and start codon are still there in the same locations as they were before. So over here, start codon ATG, that's a series of nucleotides. That's worth knowing. There's one kind of start codon that predominates over all others. ATG, and in the nucleotide version for RNA, that would be AUG. This is one of the possible stop codons. TGA in RNA becomes UGA. All right, so that's the sequence. It basically runs from AUG through all of that until stop codon UGA. And that's basically the recipe for making the protein, the TAAR5 protein. So this is one way you can get the protein sequence here. So this is the sequence of a protein. We saw the sequence of the nucleotides. And when we talk about protein sequence, what are the units in this protein sequence, right? So we got M, R, A, V, F, I, Q, G. What are those letters? Those are not nucleotides. So as you know, I'm sure the building blocks for a protein are amino acids. And amino acids, there are 20 different amino acids, and that covers almost all the letters in the alphabet. So there's quite a few different ones here. And you don't have to know them for this course, but you could look into the abbreviations. There are single letter abbreviations for amino acids and also 3-letter abbreviations for amino acids. We'll use both of those. You won't be required for this course to know really much or anything about these amino acids. But let's do this here. This is a resource where you can take a nucleotide sequence and turn it into a protein. So I'm going to back up a little bit here, and I'm going to grab this whole thing that we turned into RNA and try to translate that. So the boundaries of this sequence are the boundaries of what got turned into RNA. So this is the piece of DNA that would have gotten turned into RNA, would have been made as a transcript. So maybe a better way to say that is the RNA molecule was copied from the DNA template, was produced by looking at the DNA molecule for comparison. So I'm going to copy that and swing over to here. This is a automated translation software that's available online. And we're going to go translate. And it gives us an output that it's covering all possible situations. So there are 3 possible reading frames. We'll talk about those. And there are 2 directions, right? So the DNA molecule runs in 2 directions at the same time. We'll talk about that in the DNA lecture. And I guess what we're kind of looking for is the sequence of amino acids that covers, well, let's see, let's do more specifically. For this molecule, we're going to look at the DNA sequence, the amino acid sequence that starts first, which would reflect the first possible start codon. So this one, the first start codon is this M here for methionine, and that's way later than this molecule, which starts pretty early on. And another feature that can help us to know that this is a valid protein sequence is that it's pretty long and continuous. These other ones have stop codons buried in them, and in any individual amino acid sequence in here is not very long. So this is actually our legitimate amino acid sequence, starts MRAVF. And if we go back here, our translated version goes MRAVF. So that is the result of translating that RNA molecule. And when we get over to the mutation topic, we'll talk about how different versions of proteins can be made by having different versions of the amino acid sequence, which would result from different versions of the nucleotide sequence. So for now, we're just going to kind of talk about the topic of making this protein in general. So this would be the sequence of the protein, and the series of letters end up being the molecule building blocks that make this structure. So a lot of this is coiled. That's a notation that shows us where an alpha helix is located. And that's kind of one of the standard ways to show protein structure. So we don't need to know what proteins do or what they're, how they're put together for this class, but in like a biochemistry class, you might be interested in that. And certainly if you ever study the functions of proteins, you might get into more detail on what they're what their structure means for their function. Okay, so that was kind of our introduction to how to make a scent receiving protein. And just to zoom out to the bigger picture here, the process of making that protein is part of what we call the central dogma. So central dogma, a very big concept in biology. It's kind of in there in the name, right? Central, the central dogma. Dogma is, it's actually kind of a religious word, right? It's this thing that people really believe. The concept of dogma shows up in religion. So the central foundational concept or belief, and it's very central to biology. So it's central because DNA is the genetic information for everything that's alive. And this process of going from DNA to RNA to protein, that is present in everything that's alive. So it's very central, central to humans and fungi and fruit flies and water lilies, whatever you want. They're all using the same process. So whatever you want to study, whatever you're interested in, there's a good chance you might be employing something in this central dogma diagram for your study. So we're going to be talking about this process here, going from RNA to protein. For now, we're skipping this part. We'll come back to that later. RNA to protein, process of translation. So zooming in on those molecules, it's kind of helpful, I think, to get it in perspective how these sequences of molecules end up becoming the code for the protein. So DNA, as we talked, consists of nucleotides, 4 kinds, A, C, G, and T. DNA molecules are double-stranded, so we've got a strand on top being complementary to the strand on the bottom. And the process of transcription involves copying one of those strands into RNA nucleotides, which are A, C, G, and U. So the RNA molecule ends up being a whole lot like the DNA molecule. So we'll talk about direction in the transcription lecture, but here we've got direction 5' on the left, 3' on the right. And this strand of RNA looks a whole lot like the strand of DNA that also has 5' on the left, 3' on the right. So that's the top strand, as what we call the coding strand, CGT, GGA, CGU, GGA. So that message gets copied over into a different kind of molecule. And then the RNA molecule, known as mRNA, messenger RNA, gets translated. So the process of translation involves looking at 3 of those nucleotides at a time. And a set of 3 nucleotides is known as a codon. And any one codon is the nucleotide code for putting in a specific amino acid. So where you have CGU that says put in arginine, where you've got GGA that says put in a glycine, UAC, tyrosine, yada, 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 all the way through this sequence from start to end produces a string of amino acids that are joined together. And that string of amino acids gets called a polypeptide. Polypeptide, sometimes synonymous with protein. So we've seen different types of molecules. DNA and RNA both are nucleic acids. Protein is made up of amino acids, a different kind of molecule, a very different kind of molecule. And then as a part of this course, I like to emphasize the process that is enzyme dependent. So in your other courses, in the prerequisite course for this one, you have seen these processes and you've learned how they fit together. And you can say, all right, DNA comes from RNA. In this course, I want to start emphasizing something that might be new to you, is all the signals that are involved in this process. So there are signals in the DNA that enable transcription to happen. There are signals in the RNA that enable translation to happen. Above and beyond just what we talked about with the codons, there's lots of other signals in there that can be useful. We'll kind of cover those as we go. So zooming in on translation. Translation, as I said, this RNA, the most usual way we think about RNA is as mRNA, messenger RNA. So the messenger part of that means that it's copied from DNA. In our cells, it goes from the nucleus out into the cytoplasm, and it sort of carries a piece of information. So a message of information gets carried forward to the place where translation happens. And the protein is built by basically reading the mRNA molecule and turning it into a series of amino acids. As the mRNA gets translated, it needs some help from other molecules. And it turns out that the other types of RNA that you've learned about in other classes, they're all involved in this process. So the mRNA is the message, sort of the instructions, but the instructions get read, as it were, through a combination of these other features, the ribosome, which is made up in part of ribosomal RNA, and tRNAs, transfer RNAs, that individually each bring a new amino acid in to make up that sequence. So we'll see this part in process. The ribosome is a big molecule. It's kind of the coordinator for the whole process. So this large molecule sort of holds a RNA molecule inside of it. Sorry, I should say mRNA molecule. And it runs the mRNA molecule through the ribosome. And as a part of that process, the tRNAs come into the ribosome and they drop off their cargo. So they're sort of a carrying molecule that carries an amino acid and matches the amino acid up with the correct codon and brings it in one at a time until this entire amino acid sequence is put together. So looking a little bit more closely at what a tRNA looks like, tRNA is made up of RNA, and it has this kind of angled three-dimensional structure. So it's a single strand of RNA that's folded back on itself in what we call a secondary structure for RNA. So parts of the RNA sequence are complementary to parts later on in the RNA sequence. So you see some complementary base pairs being made here, G to C and so on, U to A. These are complementary pairings, and they make what we call a stem in this structure, stem, 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 and some loop structures. Most of that is not important for us. There's a couple parts of this molecule that are important. One of those is the anticodon. So down here at the bottom, usually when these are drawn as the anticodon, and the anticodon is a complementary match to the codon on the RNA, the mRNA molecule, I should say. The other important part of the tRNA molecule is the attachment site for the amino acid. So an individual tRNA is a specific match, makes a specific match between the anticodon sequence of nucleotides and the kind of cargo it contains. So there's an amino acid, there's a tRNA, a different kind of tRNA to carry each amino acid, and it has an identifying anticodon along with it. So part of the process of making those tRNAs useful is to load them up with an amino acid. So amino acids are not part of the RNA structure of a tRNA, and they get added. They get added by a molecule known as an aminoacyl tRNA synthetase. It's a bit of a mouthful. And this is an example here, one for phenylalanine. And so this process shows an uncharged tRNA, a tRNA that doesn't have any amino acid attached to it. And the uncharged tRNA gets charged by going into the tRNA synthetase and getting attached to a phenylalanine amino acid. So I just put the phenylalanine diagram there, but it's this little addition at the far tip of the tRNA molecule. That is the phenylalanine. And when that's attached to the tRNA, that molecule is called a charged tRNA. So tRNAs go between charged and uncharged, sort of like loading up cargo if you were a delivery truck. You get filled with the cargo and then deliver it. So deliver it to the RNA molecule. So those tRNA synthetases, there are separate kinds of tRNA synthetase. They're shown the same color here. This kind of pink color is actually the tRNA molecule interacting with the tRNA synthetase. And so on a molecular level, the tRNA synthetase recognizes the tRNA as a specific kind and loads it up with a specific amino acid. So the valine tRNA matches with the valine tRNA synthetase and gets loaded up, charged with a valine amino acid. Glutamine loaded up, now charged with glutamine, threonine tRNA loaded up with the threonine cargo and now is a charged threonine tRNA. So very much like loading up a truck. Maybe I'll do my diagram here. I kind of enjoy this one. All right, so let's imagine that we are a fleet of shipping trucks. Do my best on these. All right, we're gonna make them a flatbed. So they are, let's say we have a valine tRNA. tRNA is sort of the truck that does the moving. And when it's charged, it gets charged with the valine amino acid. Then it's loaded up and able to carry that tRNA, sorry, that amino acid that it's specific for. So this valine type of tRNA is specific to that cargo. It only delivers valine cargo. And after it's loaded up, it delivers to, where do they go? Where does the tRNA drop off its cargo? Right, the ribosome. All right, so it drops off the amino acid cargo. Then it becomes, trying to draw it in the same direction here. Then it's kind of an empty truck, but it still ships for the valine company. So now we just draw an empty flatbed here. I don't have any personal experience in shipping, so do what I can here. So the tRNA is still intact, the truck is still intact, and it's ready to be loaded up with new cargo. So load with amino acid by going to the tRNA synthetase. Amino acid. And then after that process, it is able to do more delivering. So it's going to the place where it gets loaded up with amino acids, deliver those, and comes back around. So the same tRNA molecule goes back and forth, from charged and uncharged, delivering as needed to ribosomes. So wherever a valine is called for, there would be a valine codon, and that would call in this valine tRNA to put it in there. Okay, so valine, threonine, glutamine, and so on, these are amino acids. Your book has this diagram of what the amino acids look like. In this class, I'm not particularly interested that you know really much of anything about amino acids. You don't have to know their names or their structures or their chemical properties or anything. But in case you were curious, this diagram shows you some of that, and they're grouped into categories based on their chemical properties. And the difference in chemical properties is kind of important to this class. Basically, you can put together amino acid sequences that give chemical properties to proteins. So some proteins are more hydrophobic. Proteins that end up in lipid membranes, for example, are hydrophobic, and that allows them to stay in the lipid bilayer. Some proteins have charged regions. Some are able to interact with other kinds of molecules, such as getting phosphorylated by adding a phosphate group, and so on. So it's sort of like a little toolkit. It's a really tiny Lego set of different pieces that can be put together to confer chemical properties onto the resulting protein. So these are our amino acids. The part in magenta there is how they're different from each other. And then this black part at the top, that's a feature that's in common across all the amino acids. It's sort of their linking chemical profile where they can join up to other amino acids. So an example here of a protein from fruit flies. This is the brown protein from fruit flies in one of the papers you might read during the fruit fly lab. Just as we did at the beginning of this lecture, we can see the nucleotide sequence. And this one has it broken down where the 3-letter codons for the DNA are there, as well as a single letter. Every 3 nucleotides is a letter that indicates the amino acid that would get put there. So ATG, again, the start codon brings in M-methionine, and that sequence of amino acids builds until it reaches this stop codon UGA or TGA, which has a star to tell you that's the stop codon. So throughout this sequence, there's a little bit differences along the way between how hydrophobic versus hydrophilic this molecule is. Folds around in 3 dimensions, so I'm not sure the fine details of this, but it's possible that a whole bunch of these hydrophobic regions can join together to become a hydrophobic part of this molecule. This is a membrane transporter, so it actually functions by being located in the phospholipid bilayer, so it needs to be a little bit hydrophobic or a lot hydrophobic to be able to stay in there. So it actually kind of prefers to be inside of the lipid layer, which is hydrophobic, and the hydrophobic parts of the protein fit in there. So any one amino acid here, any one of those might be maybe not so important to the overall structure, but altogether there's enough pieces that have chemical properties that are useful to make this molecule function as it needs to. All right, so the fine process of building these, I mentioned the mRNA, the rRNA in the ribosome, and then also the tRNA. Just a diagram from your book to highlight this a little bit, maybe simpler. So the mRNA molecule gets read in the same direction we saw before, 5' on the left, 3' on the right, and moving toward the 3'. The ribosome basically clamps down on the mRNA molecule and zips through it, scanning for nucleotide sequences of codons where it finds the first start codon. That's the start of the sequence, and then it keeps going along and adding on amino acids as it goes. So it's going to keep going toward the 3' direction until it hits a stop codon. So here's a little finer detail of the interaction of one codon with one anticodon. So the codon is present on part of the mRNA molecule, and the anticodon is present on the tRNA molecule. And this codon, GUU, matches with the anticodon CAA and brings in this valine amino acid. So some fine details in the text here. We talked about the different molecules that are involved. The start of translation beginning at the first start codon. So not necessarily the first nucleotide of the mRNA molecule, but the first start codon. And then a little bit about this interaction. This tends to throw students a little bit. The mRNA molecule has the 5' on the left and the 3' on the right. Put that on there here. Draw it out here where it shows up better. 5' on the left, 3' on the right. That would be the mRNA molecule. Then the anticodon is actually anti-parallel to that. So at the location of the anti-parallel anticodon, it's actually 5' on the right and 3' on the left. So this question that's being asked on the third bullet point, in what direction does the anticodon run? It would actually run, if you had the 5' on there, it's actually 5' AAC, right? So just kind of a reminder, this is how nucleotides recognize each other with these complementary interactions. Whether it's DNA to DNA, DNA to RNA, or in this case, RNA to RNA, they all obey the same rule about matching each other as this anti-parallel running in opposite directions interaction. Okay, so once translation, once translation starts, the start codon here, AUG, then it continues going in the toward the 3' direction on the RNA molecule, mRNA molecule. Don't worry about these E, P, and A letters. I don't think that's so vital for our class. But it starts methionine, and then the next 3 letters. So whatever 3 letters follow the first codon, those become a codon. You'll notice there's not really a natural break between any of these letters. C, 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 A, C, G, they're just letters. But knowing that they should be read 3 at a time, that's the job of the ribosome. So the ribosome has these positions in it that hold the mRNA molecule in a place where it can be interpreted and joined to the corresponding tRNA. So every tRNA is on there immediately after the one before, and that means the next 3 nucleotides after these 3 will get read as the next codon. So AUG is a codon, then CCC is a codon. And that ends up being something we call the reading frame. So within a nucleotide sequence, the portion that has the coding sequence, the part that codes for protein, goes 3, 3, 3, 3, 3 nucleotides all throughout from the start codon all the way until the stop codon. So a little bit about our codons here. These are all the possible codons. And of course, if you've got 3 nucleotide letters and 4 possible nucleotides at each position, that gives you this grid here. So there's a first letter, first base, for second base, third base, the 3 letters that make up the codon. There end up being 4 to the 3rd power. So 4 times 4 times 4, or 64 possible combinations of codons. So those are all shown here. A few of them are highlighted in different colors, including the start codon, which is AUG for methionine, MET amino acid. And then we have 3 kinds here in this magenta color. These are stop codons. So if the ribosome encounters a UAA, UAG, or UGA, then that stops the amino acid sequence from being built. Basically stops it wherever it got to at that point. One of the interesting properties of RNA, you might have seen it before on the structure of the tRNA molecule, RNA gets away with a base pair that's a little bit unconventional. It's called a wobble pairing, and it's acceptable in RNA-RNA interactions. It's not a great bond, but it's an acceptable chemical bond to match a G nucleotide against a U nucleotide. And in the process of translation, that means actually is allowable only at this 3rd position. So the 3rd codon position is allowed to do a little bit of this wobble pairing. So in this example, shows the same tRNA molecule, right? So this tRNA bringing serine has the same anticodon, AGG, AGG. This AGG anticodon is able to match UCC and also UCU. So A matches U, G matches C, and G is okay matching with U according to the wobble pairing. So what that means is that the same tRNA can do double duty for quite a few of these amino acid, the codons. So notice how they're paired up. We've got the codon here, and then in the text to the side, I've put the anticodon that would be able to, for many of these, do double duty to match up to both of those. So phenylalanine UUU or UUC, AAG is a proper match for UUC, but AAG is an acceptable match for UUU, right? G to U in that 3rd position. So the way this lines up is in a block of 4, the top 2 can match with one kind of anticodon, the bottom 2 can match with another kind of anticodon. And that's pretty true throughout. There's a few exceptions here. Methionine is very specific UAC anticodon, so not a wobble pairing here. And then some of our stop codons are very specific matches that are not the same as the wobble matches. So what this means is that there can be a lot fewer tRNA molecules than there are possible codons. So I mentioned there are 64 possible codons. The actual number of tRNAs is a little tricky to say just universally. There's some variation in the tRNAs. But on this image here, I've got roughly about half. So just a little over half of the total number of codons need to have perfectly matching tRNAs in this situation. So I guess you could think cells are a little clever, or at least efficient. Let's go efficient and not clever. Efficient on a molecular level, that they can get away with less. So they don't need 64 anticodons if they can do the wobble pairing. So again, the concept of wobble pairing is important. I just want to sort of illustrate how that works with a genetic code, but you don't need to know the fine details of any one of these codon, anticodon matchings. All right, so going back to our sequence there, we could zoom in here and do the translation by eye using our translation table. The translation table is written according to the codons and expecting this 5' on the left, 3' on the right direction. So I've highlighted start codon, stop codon, start codon, AUG. Turns out it's the first start codon. So you could scan through here and say, is there any AUG as I go along? Don't see it, 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 don't see it. Oh, okay. So this is actually the first of this sequence of nucleotides, is the first start codon. So I trimmed it down quite a bit. I left you a little bit of the first line and a little bit of the last line. So you could go in and find AUG corresponds to methionine. And then according to the reading frame, the next 3 letters are AGA. So we look on here, AGA, it goes for ARG or arginine. Then GCU, dun, 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 GCU, alanine, and so on. So arginine, alanine, valine, phenylalanine, and so on. And it's only when there's a 3-letter codon that matches the stop codon that this process actually stops. So after the stop codon, it doesn't matter what these nucleotides are, they're not going to get translated. Translation is done for the day at that point. So this would match up with the sequence we saw earlier. We had before the single nucleotide abbreviations. There we are. So it started MRAV, and maybe take my word for it. M for methionine, R for arginine, A for alanine, V for valine. So it is the same sequence automatically generated and also what we could translate here. There it is, MRAVF, and so on. All right, so final thing we want to talk about here is how this amino acid sequence kind of works into the 3-dimensional structure of the protein and also its function. So there are these terms referring to the levels of organization for a protein. They start off as the amino acid sequence, as we've just done. That's what's known as the primary structure. But of course, molecules don't just exist as these linear molecules. They end up folding up in 3 dimensions. So they have some secondary structures, such as those coils we saw before. This is an alpha helix. This is commonly found, so it's just energetically more efficient to kind of pack up this molecule. And ultimately, the whole series of amino acids take on this 3-dimensional structure. So this 3-dimensional structure is called tertiary structure. So we just have primary, secondary, tertiary, quaternary. These are first order, second order, third order, fourth order structures. And there's this concept of quaternary structure is in a protein that might have more than one what we call subunits. So the tertiary structure, difference between tertiary and quaternary structure, the tertiary structure is the result of translating one mRNA. But the tertiary structure protein might not have a final function. Sometimes it's a subunit and it has to be packed together with other subunits to make a functional protein. So not every a protein is just a one-to-one, one gene makes one protein. Some proteins are made up of two or more genes, as we will see. So it says here, not all proteins have quaternary structure. Some proteins end up making just a single, or sorry, some genes end up making just one protein as a result in this tertiary structure. So some examples here, this is going back to the brown protein from fruit flies. This is a final protein known as a heterodimer. So the dimer part refers to the fact that it's made up of two units. And hetero part refers to the fact that it's got two different genes making up those units. So the brown protein is part of this transporter called an ABC transporter. And brown is one half, and this other gene called white is the other half. And they're actually pretty similar proteins you can kind of see there. So brown plus white, those two separate proteins together, each with their own tertiary structure, combine together to make a quaternary structure, which is the functional protein. So the individual gene products would be called the brown protein and the white protein, and the functional transporter might be called the ABC transporter. So heterodimer example, also possible to have a homodimer. Homodimer consists of two subunits, but each subunit is made by translating the same gene. So this is two copies of the same protein put together, each one produced by this ADH5 gene, and they work in a pair, they join together, and then they have a function when they're paired together. If you're curious, this is a molecule that helps people to detoxify alcohol in their system. So alcohol dehydrogenase. And then it just keeps getting more complicated. If you look up some of these more complicated proteins, this is one that's very important to life. This is the ATP synthase protein. In the process of cellular respiration and photosynthesis, this is a molecule that generates ATP using a proton gradient. Maybe you remember that. And all the different colors on here are different protein subunits. And I've highlighted just a few of them, not even all of them. But the ones I've highlighted, I've shown you what the name for the gene is and where it's located. This is a human version. And so within a human, we've got examples of chromosome 18, chromosome 12, chromosome 19, 10, 20, 17. These are pieces of genetic information coming from all over the genome and getting assembled on the back end into a functional protein. So just kind of amazing sometimes what it takes to put together one of these big functional proteins.